Peace be upon you, grade 9 students. I hope you are well. In this video, we are going to study Chapter 1, Activity 4 from Foods to Nutrients, Part 1. Before we start, prepare these materials: a notebook and a pen, your support guide, and a phone or laptop. And please follow these instructions. At the end of this video, you will be able to list the organs of digestive system, distinguish between digestive system and digestive tract, analyze experiments of in vitro digestion of some food types like proteins, identify the role of bile, represent or describe the molecular simplification of some food using a functional diagram. Most of the food that we consume is made up of complex molecules like carbohydrates, proteins, and lipids. Upon digestion, these complex molecules are broken down into simpler ones. Where and how is the food transformed into simpler molecules? Of course, this is the function of the digestive system. Let's recall the organs of the digestive system. First, we start by the mouth, then esophagus, stomach, pancreas, small intestine, large intestine, and the liver. Sometimes people confuse between the digestive system and the digestive tract. Well, they are totally different. The digestive system includes both the digestive tract and the digestive glands. The digestive tract is the pathway of food. It's the tube where the food passes throughout the digestion. It includes the mouth, esophagus, stomach, small intestine, and large intestine. While the digestive glands, it includes the organs or the glands that secrete the juices necessary for the digestion process. It includes the salivary glands, gastric glands, liver, pancreas, and intestinal gland. So, to sum up, after ingesting the food, it starts from the mouth to the esophagus, then the stomach, to the small intestine, and finally ends in the large intestine. These are the organs of the digestive tract. So, the food passes exclusively in the digestive tract or tube. Food never passes into the glands like the liver or the pancreas. The glands secrete the juices that ensure the chemical digestion of food. After having studied the transformation of starch by salivary amylase, let's find out what happens to other types of food like proteins. But before we study the digestion of proteins, let's learn about its structure. Proteins are complex molecules made up of several peptide chains. The peptide chain in its turn is made up of a sequence of amino acids, the simplest molecule in proteins. Food in the stomach is mixed with the gastric juice, and the gastric juice contains an enzyme called pepsin. The gastric juice also contains hydrochloric acid HCl that renders the medium acidic for the optimal activity of pepsin. So, the gastric juice, which is constituted of both the hydrochloric acid and the pepsin enzyme, they accomplish the chemical digestion of proteins into peptides. And the chemical digestion of proteins is also facilitated by the mechanical digestion, which is favored by churning or the muscular contraction of the stomach walls. Then, pepsin acts in acidic medium. The pepsin hydrolyzes the complex protein molecules into smaller ones called peptides. However, peptides need further digestion by another enzyme in order to be transformed into the simplest molecules. Where do proteins continue their digestion? What are the glands and the juices involved in this digestion? Digestion, a passage from complex to simple. In order to answer these problems, we conducted an experiment where a tube containing coagulated egg white and pancreatic extract is put in a water bath for 1 hour. The substrate in this experiment is chosen to be the coagulated egg white or protein. But what do we mean by coagulated egg white? Coagulation is defined as the change in the structure of protein from a liquid form to solid or a thicker liquid brought about by heat. As we see in this figure, the liquid part of the egg, which is the egg albumin, changes from the liquid state into the solid state upon heating. This is the coagulation of albumin. Note that only proteins coagulate. Peptides and amino acids don't. Also note that proteins and peptides give a positive result with biuret test, however the amino acids don't. As we see here, the proteins and the peptides give a positive result with the biuret test, and which is revealed by the appearance of the violet color. While with the amino acids, there is no appearance of violet color, so the initial color of the biuret solution appear, which is blue. Let's come back to our experiment and interpret our experiment. Remember, when you are asked to interpret, so you must make analyze plus give a significance. After 1 hour, the tube containing coagulated egg white and pancreatic extract became clear. This means that the egg white is digested. This statement represents the significance. What do you conclude from this interpretation? The egg white is digested. This means that the egg white disappeared. Let's observe what do we have inside the test tube other than the egg white. We have the pancreatic extract. So, draw out a conclusion. The conclusion is the pancreatic extract contains an enzyme that acts on proteins. Contents of the pancreatic juice. The pancreatic juice contains 3 enzymes: trypsin that acts on proteins, lipase that acts on lipids, and amylase that acts on starch. So digestion of proteins continues in the small intestine under the action of trypsin. Enzymes that act on proteins are called proteases. So both pepsin and trypsin are proteases. But in the contrast of pepsin, which acts in acidic medium, trypsin acts in basic medium. This graph shows that the optimal activity of trypsin is at pH greater than 7. So the trypsin acts in basic medium. How is the medium rendered basic in the small intestine? The answer is that the liver produces bile. This is the liver and it produces bile. Bile is a liquid that contains no enzymes. Bile is stored in the gallbladder and conducted into the small intestine. So, the bile is produced by the liver and it's stored in the gallbladder and it's conducted into the first region of the small intestine. This figure shows how the bile is conducted into the small intestine through the bile ducts. It renders the medium basic for the optimal activity of intestinal enzymes. Application. The adjacent figure represents an experiment that aims to study the digestion of a piece of meat in presence of pancreatic juice. This figure shows the presence of 2 tubes A and B. Both tubes contain a piece of meat plus pancreatic juice, and they are put in a water bath at 37 degrees Celsius. The only difference between these 2 tubes is in the chemical medium, where in tube A, pH equal 8.5, while in tube B, pH equal 2. Let's start by the questions. Pose the problem studied in this experiment. Remember that the problem is a scientific question and it must end by a question mark. What is the suitable chemical medium for digestion of meat by pancreatic juice? Name the protease present in the pancreatic juice. It's what? Trypsin. Construct a table showing the experimental conditions and the result of the experiment in tubes A and B. In order to construct this table, you must mention all the conditions or all the variables which are found in the experiment and the results obtained from this experiment. The table below shows the experimental conditions and results at the end of the experiment in tubes A and B. This is the title of the table. And this table, we have the substrate, which is the meat in both tubes A and B. The juice used is the pancreatic juice, also in both A and B. The temperature is 37 degrees. The time or the duration is 60 minutes. The only difference between them is the pH, where pH in tube A is 8.5 while in B is 2. And the result with the biorit test is the appearance of violet color in tube B while the presence of the blue color in tube A. Interpret the results of the experiment. Always try to start your interpretation with the results of the experiment. After 1 hour, biorit test gave a blue color negative result in tube A where pH equal to 8.5. This indicates that there are no more proteins in this tube. However, biorit test gave a violet color in tube B where pH equal to. This indicates that proteins still exist in tube B. Derive a conclusion concerning the tube where digestion took place. Digestion took place in tube A where pH equal 8.5. Determine the biorit test result in tube B if the pancreatic juice is replaced by gastric juice. Remember that in verb determine, we start by logical reasoning depending on our acquired knowledge and the given information, and then we give the answer. Think, which enzyme does the gastric juice contain? And in which chemical medium does the gastric juice act? Since the gastric juice contains pepsin, which acts in acidic medium, then the chemical medium in tube B is suitable for the pepsin activity, which is pH equal to. Pepsin hydrolyzes proteins into peptides, so the biorit test result will be positive and it will give a violet color, since the peptides give a positive result with the biorit test. Draw a functional diagram that shows the hydrolysis of meat that took place in tube A. First, we represent the structure of the protein. The protein is digested by trypsin to produce peptide, and then the peptide is further digested by trypsin to produce the final product, which is the amino acids. You must write your legend where this arrow used in the figure represents the word produce. Of course, we don't forget the title. The title is a functional diagram that shows the digestion of meat in tube A. Summing up, the digestive system is made up of the digestive tract and the digestive glands. The digestion of proteins starts in the stomach, where the gastric gland produces the gastric juice. The gastric juice contains the pepsin enzyme. The pepsin enzyme transforms the proteins into peptides. Digestion of proteins continues in the small intestine. The liver also produces the bile. The bile makes the chemical medium basic. The pancreas releases the trypsin enzyme. The trypsin digests or transforms the proteins and peptides into amino acids. You will be assigned to copy and memorize the summing up, Solve questions number 11, page 12, plus number 15, page 15 on the support guide. I hope you enjoyed the lesson. Keep safe.